So it was really nice to see that there is a transcriptome difference in these cells from Himena, because that's how our venture into all these studies started, when we look at patient skin cells and we discovered that there was a gene expression difference. So when we look at all these neurons and all what Paul was showing and what Jean-Michel was showing and others, it would be really good to see how these different colored cells also look in terms of gene expression. So we were pursuing that to a large extent. In my talk, I went back to the part 2 of separating wheat from the chaff, where what I mean by that is just looking really what's important and perhaps what's less important in looking at DNA variation, understanding of its impact and how it's actually important in a human situation. In my life, I love the models, but I really, for the sake of at least having focus, I'm really very focused to work with patient material and patient-derived primary material in terms of relevance to study a particular disorder. So that's because that maintains really the complexity which lots of models perhaps cannot capture on purpose. I have a little disclosure. I'm working with Marinus in terms of advising them on some of the aspects of this study, and I'm also sitting on the PCDH19 Alliance scientific board. Thank you. So I would quickly just recap, because is it almost 1 year and 2 days since we published in 2008, 1 year, 10 years and 2 days since we published the PCDH19 paper. It's actually appeared in the June issue of the journal in 2008. So it's a decade ago, and it's quite amazing how much happened really in that space and that time. So we found it as a cell adhesion molecule, and you know that this molecule is important for axon bundling. So where you saw those stripes, a lot of them represent bundles of axons which are similar and via the brain. It is a synapse protein, we heard, and that's now pretty well validated. It is a molecule which interacts with important GABA molecules, like subunits of the GABA receptor. It's the molecule which feeds back to the cytoskeleton, and there are several other molecules which have been shown to interact. We're not necessarily following all those cues very actively, but this is not a unique feature of protocadherin 19. Interestingly, there are upstream regulators of PCDH19, and one of them is microRNA 484. And it's interesting that this is from the region of another neurodevelopmental disorder, which is frequently deleted. And through that microRNA, it's actually PCDH19 is involved, which tells you that PCDH19 is not a disorder of importance for its own, but perhaps for a broader range of similar seizure and learning and behavioral disorders in what we know. There were multiple now knockout models generated. There are some fear response and hyperactivity phenotypes, which we found to some extent also with Paul, but we were not that happy about all the robustness of those tests, but there was some indication. We reported on that allopregnanolone, and later on with Nicolas Pecchio, also cortisone deficiency, and this was endogenous because with the ACTH stimulation we found that these girls had this sort of mechanism of stress response, if I put it that way, severely compromised. And I would like to say that it does look like PCDH19 is probably the most prevalent epilepsy gene or disorder in girls. Now I'm using the acronym GCE. We are responsible for the acronym FLE, but I think 2 years ago we agreed, at least most of us, that we're going to be using girls clustering epilepsy, so I'm just sticking to that, at least for the time being, unless somebody like the nomenclature committee of Ingrid Scheffer is going to come up with something better. So today I would like to tell you, because it's quite important, about when a DNA variant is important or whether it's not important, especially in the era where almost all of us are eventually going to have our genome sequenced, and we have a lot of changes. So I go a little bit back and have some general information or reflections. So why we are different. Of course, we can find answers like here. We have tallest and shortest men in the world. We can find the answers in perhaps nutrition. We know that. There are differences in height and latitude. It doesn't matter whether you live in Italy or whether you live in Denmark. Even within the same country, there are body height differences. And you know that hard work also affects the way how you're going to end up. So there are genetic and epigenetic causes of the differences in traits, what we see. And that's why we study DNA. Now, when you look at the human genome, there's about 3 billion letters. In my speed, probably would take me 200 years to read a human genome from beginning to the end. But if you were reading 24 hours and you were speaking about this in my space, it would take you 100 years to read it all. Now, what is interesting that if you were going to read only the differences between the average two of us in a room, you would need to read it in that pace for 4 months, non-stop. So that's the number of single-letter differences between an average two people in this room, but there are more differences, and some of them probably haven't even tapped into. So it's extremely complicated, not talking about the number of cells, what we have, and each one of them, almost, apart perhaps some red cells, they contain the human genome. And that has to be copied from cell to cell as we grow, and quite accurately. But the last point is, which is relevant to PCDH19, is the fact that we say that our cells are genetically identical, but in fact they are not. Well, for the purpose of the textbooks exercise, they are. For the practical purposes of intervention, like in a disorder like PCDH19, we have to admit and appreciate that these cells are not identical. Like in females, you know that you are kind of genetically identical, but you are mosaics simply because you have X inactivation. So you have two sets of cells. Depends whether you have maternal or paternal chromosome X active. And that technically gives you actually a great advantage against males, because males have only one X chromosome. So you have two solutions. You have two solutions to a problem, while males have only one. So this is actually a highly important evolutionary phenomenon which we are also studying, while females are actually generally very robust to genetic mutation. It's just female tolerates. Like, you can be a female, you can have a genetic mutations, you can pass it on into your male offspring, and they are affected and you are not. And that simply can be because you are female. So this mosaicism, which is generally an evolutionary protective, it's clearly in PCDH19 is the trouble. So it's really very important that we get to the bottom of it, of what is driving it and how we can address it. What you heard from Kimina and the others, the gene expression in PCDH19 cells, what we found, they are very different, but for the purpose of the exercise, it's normal because the males with the mutation PCDH19 are not affected. So it tells us sort of how extreme that can be, but we also need to consider these gene expression differences in the context of the male-female situation. So going into this, we have the situations, of course, in PCDH19 where these variants are passed on from transmitting male into affected daughter. Now we see more and more cases like that where the variants are transmitted from non-presenting mom into presenting daughter. And we have also instances where the variant is either completely de novo or it's transmitted from a father into, for example, monozygotic or identical twins, and only one of them is affected, and the other one is not. So we're collecting this information, we're trying to look into it, try to understand what's going on, and that's again one very important sort of direction in the study of my laboratory, where we're trying to understand what actually modifies the presentation of genetic variation under these conditions. It's not necessarily because you want to make a diagnosis. It's important we can make a prognosis, because I believe that in the future, maybe our children or grandchildren will be sequenced at birth for many good reasons. But then there are going to be these uncertain variants. Well, how are we going to tell whether somebody with PCDH19 at this day 5 of birth going to have a PCDH19 variant, are they going to end up like this one, or are they going to be like this one? Genotypically, they are identical. So how are we going to say? And now we know there are 5,000 different conditions. We have 5,000 genes for different traits where we perhaps can do this. I'm not talking about cancer and other elements. So we do study these really difficult questions of genetic pleiotropy or polyphenism, where really PCDH19 is a prime example, and we look for examples of DNA, RNA at proteins levels. There are many different ways how that can be altered. Of course, can be the environment, which can be the epigenetics. It is the X inactivation, eventually. Environment is not always the outside environment. It can be the environment of your genes, can be the intrauterine environment, something which is in addition to genes. And we have something also like a polygenic risk. That's the kind of the risk of all the other genes which comes together with the variants, which we need to actually understand, because that can explain up to 10% of the variation, what we see. So we do study this dilemma. You put in a particular variant and you get different outcomes. Now, these data are updated. So we really, in PCDH19, see the clusters at about up to 95% of cases. Cognitive outcome, about 70% of cases. Autism, 20% of cases. Behaviors, 65% of cases. And the penetrance, it's pretty much or reduced, or we can almost call these individuals a non-penetrant, up to 20%. So 1 in 5 carriers of PCDH19 variant, which will meet the criteria of deleteriousness overall, is not going to present. Well, that's a complication, because it's a complication for intervention, it's a complication for diagnosis, prognosis, but at least we're refining it. Our original studies indicated that, based on families 10 years ago, that this non-penetrance was maybe 5 to 10%. I think we are now pretty sure it's 20%, and simply because the ascertainment of these individuals changed. We don't track them through the families, which is biased, but we really track them from the population, because the technologies of DNA investigations are being much more accessible. But they are definitely not widely accessible, as one would say. So that's probably where we're going to see probably a lot more, and I would say the penetrance probably will go even further down if you're going to just systematically investigate larger cohorts. So we think the X inactivation is a major player, but there probably can be others. So that's why we really said, before we run into all these sophisticated experiments, or while we're doing it, we should probably look at the new data since the discovery of the gene, because over 10 years there can be a significant amount of data which can inform us which way to move, where to look, or perhaps where not to look. So with Christy Colts, who is a really very young, dynamic lady, got married last year in December. She's doing PhD with us. Her background is in psychology, and she was attracted by this topic simply because of the complexity and the importance of genetics as well as environment. We decided to perform a review, and as well as she suggested, so we need to design a more detailed and very specific survey, look into the behavioral phenotype in these girls, because that started to really become obvious to us. It's becoming the second, if not the most important presentation in this group of disorders, in addition to seizures. So we review all the literature. There were about 320 cases altogether at that time, which is probably goes about 4 months ago when we submitted the paper. The paper should be online pretty much any day, Monday, Tuesday. We corrected the proof, so you will be able to access it very quickly. But when we looked at independent cases and removed all the duplicates, so we had about 271 cases. I can tell you since then there are probably another 100 published. So that tells you how many have been published. And by knowing how it works, that probably it's easy, twice as many already diagnosed. So we're reaching really 500, 600 cases which are probably already diagnosed in individuals who live in families with this disorder. Our major finding was, going really to the nutshell, was the earlier you have the seizure, the more severe outcome you get. The mean onset of seizures was 11.9 months, and the median onset of seizures was 10 months. But if we stratified simply based on the 12 months, if you're born, or if you have the first seizure before or within the first 12 months, your outcome or your prognosis was much worse than if you had a seizure after the 12 months. There are exceptions, but generally this was very strong. And you will see the scale on the left is mean ID severity. I mean, we didn't do it as a continual measure because that's based on some different reports. Sometimes they report IQ, sometimes they don't. So we reclassify the intellectual disability for 0, 1, 2, 3, 4, 5 from no, borderline, mild, moderate, severe. So in a way, the difference is almost 1.5 steps. So generally, before 12 months onset, you end up with borderline, no, borderline clinical cognitive outcome. But if you have it before 12 months, you end up actually with quite severe to profound. So that may have some implications the way how these patients might be counseled, but it is just a reflection of this number of cases that's again going to be refined. Now, we try to capture it in this graph, which is, of course, looks complicated in the first glance. So it's one of the circus plots. So here, if you split it vertically, on the left, you have the onset before 12 months and after 12 months, and the proportions of that, I think, gray and black area just tell you the number of patients, simply. So it's in its proportions. And then on the right-hand side, these gray and black areas shooting into different levels of intellectual disability, as we coded here for the purpose of the graph. And you see that really after 12 months onset, you have very few lines going to severe or profound. Just to illustrate, I don't orient you so you can understand that. Now, on top of it, it's complicated because we put 2 additional colors, and that is the red and the blue. And these 2 actually correspond to these 2 most recurrent variants in PCDH19. And that's, one is the missense change, and you saw some results Paul showed also in his presentation, which is the variant which completely abolished the cell adhesion. And the other one is the frameshift variant, and I think also we have seen some stem cells generated. For the missense variant, which is the red one, you can see it from here, we have about 25 different individuals from about 20 different families. So it's highly recurrent. So it happens again and again. So this is not something historic which happens 1,000 years ago and we just get it. So it happens again and again. And the same for the frameshift. But then you see how the blue and the red are shooting again from before 12 months onset and after 12 months onset into different areas of intellectual handicap outcome. They go all over the place. So in a way, here you have exactly the same genetic insult, which gives you very different outcomes. So if somebody showed this to me 15 years ago, I said, this is not genetic. I mean, this has nothing to do with genetics because it's like a lottery. If you toss your coin, you end up almost with the same outcomes. But of course, that's not true. This is just a complication of the PCDH19 situation. But what is really interesting on it, we don't know, that you don't see the red ones shooting into very severe. And there are very few ones going into moderate. So it looks like that the total loss of PCDH19 might be more severe than the missense variant, at least based on this. For the time being, we say all these mutations are the same, all these missense changes are loss of function as much as complete deletion of the molecule. But I think this data perhaps hint that it may not be that simple, and maybe we just need to have more and dig into it. This is a little bit more in detail just to show you all those patients, type of seizures. I'm just trying to make some of this bigger. I'll go back because I can't read it on the screen that well. and give you the outcome where you see the vertical errors from really very normal to mild, moderate, severe. This is exactly the same variant, even in some the same families. So why we see such a variable outcome, and how we can put this into really our strategies to manage it. Now, overall, just to give you a snapshot, so there were about 145 unique ways how PCDH19 can be affected. Majority of these are in the extracellular part, so that's the part which is in the green boxes, so that's the part which is sticking out of the cell. So that's the extracellular repeat regions, which the coherent repeats. And you see below the number of dots, they are in different colors because they just reflect missense, nonsense, and frameshift variants. But it's also interesting that majority of them, actually 50% of all those variants, were actually in the extracellular coherent domains 3 and 4. Now, what is very specific about them, I don't know, but they are probably the strongest points of the adhesion when these coherents shake their hands on the opposite cells. And maybe that's why we see some enrichment. The two variants which I talked about are here, and the size of the lollipop here really reflects the recurrence. So the bigger the lollipop is, many more individuals with exactly the same variant have been identified. Now, you also see that about half of all these variants is de novo, so these are singleton cases where this error happens in that individual for the first time. And what that means that 50% are inherited, so we have them in families. And again, that's going to probably grow because we're going to have a more non-penetrant cases, and they will be always ascertained via affected daughter, and when you track it in a family, you see that probably father has it, or more likely, what we see more often, the unaffected mum has it. In terms of other comorbidities, we found 60% to 65 psychiatric autism, I mentioned that. ADHD and hyperactivity were very frequent. Now, these percentages are fairly low because not everybody reports on it for the time being. So that's, I would say, under ascertainment. And there are other multiple comorbidities. Interestingly, 25% of individuals with no cognitive involvement had actually psychiatric and behavioral problems, which is also important to realize that this is not only seizure disorder. This is a lot more complex, and I guess that starts to transpire. Christy designed this very complicated survey, which we don't have data on, but we're looking at different sort of aspects of the behavior of these individuals, and I would like to thank every one of you in the room who completed it and helping, and I know Julie and many of you have been doing an excellent job, and we're just delighted that we have such a good response rate. I will get to it. But we're trying to really address the sort of the autism side of it, ADHD, the OCD, obsessive-compulsive disorder, or tendencies, as well as the epilepsy side, as well as the mutations, some of the drug treatment successes or not, and other demographic data. So we really try to look not only at girls who have this mutation and have problems, we try to look at the parents and caregivers. We collected actually quite a few males who are mosaic and presenting, but we have some mosaic males who are not presenting. There are some around, so it is the level of mosaicism which affects it. It is the stage of postzygotic development when these mosaicism kick in, which we of course can't look back, but we can somehow deconstruct based on the accessible tissues. The survey is available in different languages, and Christy just emailed me that it's available in French if anyone would like to take the challenge. But in France, we have a large collection of families, so we decided to translate it into the French, and we are close by almost to translate it to Spanish, but not yet. So for the time being, it's in English, Italian, and French. And it's not available for children younger than 2 years old for the reasons of the design, which I really can't comment on in great detail. So we have done 80 completed surveys, which is fantastic, from 130 sent. So the response rate, I'm told, it's very good. We have 10 males 70 females, and we have 9 females which are unaffected. This is also very important to really get that information about these as much as we can. And interestingly, there is other benefits, and this is only one of them. We identify 15 novel variants which are not published in the literature. Now, why we are not just happy with the review, why do we need to do the survey? Well, I believe in the scientific publications and all of it and integrity. I believe we publish cases which are different, and we not necessarily publish cases which fit the best and most frequent description, because editors are not going to be interested. Our scientific colleagues are not going to be interested to publish another case who is the same. I mean, they tell me, well, what's the novelty here? I mean, that's the first criterion for publication. So we need to capture, and that's what registers do, and that's another aspect. The survey is not a register. It doesn't mean to compete with the register. It's really mean to, in a perhaps less onerous and faster way, or a more specific way to address aspects of a specific phenotype. So the results hopefully will be ready by the end of this year, because Christy has to finish her PhD, so you will hear about it very soon. The other area where we really pushed a lot was the interpretation of the variants, and Yuyan has been very important. And I think this is important for you here, precision medicine. I think really it's a HD medicine. It's a high-definition medicine where it's not just the precision, but the whole holiness of the medicine which comes around with the proper diagnosis and care in a timely and very specific manner. So this is the pile-up of the latest number of mutations. As you can see, there's a lot of them, and they are all in the extracellular and some in the intracellular region, mostly the frame shift and stop. These are all the variants, about 40, which we tested. We went to a lot of extra trouble. Now there are probably about 50 different tools, computer-based in silico tools. We could test, we didn't test all of them. We make a list of representatives. What also changed from 10 years ago is that now we have reference populations. So this clearly tells you how beneficial it is to share information, what you do through your parents' associations, what we try to do in genetics, put a lot of this information in for public good without the risk of identification and harm. But like Moncol-Leggett and MacArthur, our Australian colleagues now working at the Broad Institute, really released not a long time ago these reference databases which tell you about variants in different genes from up to 130,000 individuals from different ethnic backgrounds, because we need to understand that together with our variants of in terms of meaning to a specific, sometimes ethnic group. Also for PCDH19, we have no reason that the ethnicity would be any major modifier for the time being. So these are some of the stats. So we looked at about 238 frequent variants from these databases, where we have, I guess, the reason to speculate these are probably not really causing any trouble. And then we extracted about 73 variants from which were identified in the girls with these epilepsies. And we focused specifically on missense variants, because these are the hardest ones to really assess in terms of their meaning. And when we try the tools, and these are the most successful tools, so pretty much what we're looking at, the bar moving well away from these two, especially from this one, because these are the kind of frequent variants where there's no reason to speculate that they will have an impact on the function of this coherent. We tried in different ways to visualize it. Here you can see a two-dimensional space, the two different tools, but pretty much what you're looking at in the top right corner, you would like to see all the blue ones clustering, and all the green ones should be somewhere here, which pretty much they are, and these are all the blue ones. And now we tested in different combinations, permutations, and now we have a new set of these variants where we can really predict how well our tools are performing. In addition to it, in silico tools, we use our experimental tools where we use the protein structure, because fortunately for at least the zebrafish protocoherent, some of the extracellular parts of the protein have had crystal structure determined, so we actually can predict what is the effect of some of these variants, which my colleague did. And then we went to an extra trouble to actually do these estrogen receptor reporter assays, where we actually can test variant by variant, one by one, and compare it actually to what we have seen in skin cells from the girls, which we have about 30 different skin biopsies from different girls with these variants. And we can very robustly assess the gene expression as a marker of deleteriousness of these variants. So this is just a bit of a small picture to show you how the extracellular cadherin domains, how they fold, where are the sensitive spots in terms of glycosylations or calcium binding, which are very important molecules for function of this protocadherin as a cell adhesion, and especially homotypic cell adhesion molecule. Now, so these are our receptor assay, estrogen receptor. Now, this is something I wouldn't believe, that protocadherin cell adhesion molecule regulate gene expressions through something like estrogen receptor. And it's still puzzling us, and puzzling us still something which we haven't really followed, and I haven't heard here mentioned, that in the skin cells of these girls, we originally discovered that the gene expression switched from girl-like expression more to boy-like expression in this cell type, because, of course, it doesn't mean that there is a reversal from girlness to boyness, but it is in that cell type the gene expression was completely shifted from one to the other, and that's how we came to estrogen receptor signaling. And that still might be relevant in terms of estrogen receptor function, because estrogen receptor works differently in males and females' brains, and consequently these cells may have a different gene expression pattern, which we believe it's part of the picture, not necessarily the wiring and connectivity, but also actually the functionality of these cells in response to the stimulus. So we use that system, and it's pretty robust. When we put all the bits in together, we do it always with estradiol, which is the estrogen. And when we test the wild type, we see a great response. As soon as we put a range of variants where we have evidence that these cause the problem, we see a loss of function. When we pile it up across all different tools and different types of variants, we can test, and we tested actually experimentally across all of them. We can very accurately predict the function of the variants which cause the epilepsy as we know them. The variants of unknown significance, the orange one, VOS, there are 4 out of 10 behaves as pathogenic. So I can't tell you very much about these, because these variants were ascertained from large epilepsy genomic studies like EPI4K, where perhaps we're linking PCDH19 to a different types of epilepsy. Depends on the variation. So again, I'm alluding to the fact that PCDH19 may not be just a disorder as you know it. PCDH19 might be broader and not necessarily involve clustering epilepsy, but no epilepsy, just behaviors or other type of epilepsy. Where we perhaps struggled a little bit, but again, it's hard to say whether we struggled or we pointing into something interesting. The frequent variants, we generally wouldn't believe that these ones are major drivers of anything. They are just there because they are tolerated. But in 1 out of 3, we kept getting a signal. This is not very frequent variant. It's present on 1 in 500 or 1 in 1,000 individuals. It's reasonably frequent. But the question is this really an innocuous variant, or is this a variant which is predisposing to some kind of neurobehavioral problem, which we don't know. But clearly from all these, when we put it together, that was the only one where we perhaps struggled a little bit. And on top of it, of course, we can put also the American College of Medical Genetics and Pathologists guidelines, and then we get really very good readout, again, apart from the very frequent variants, which may be just the fact that some of these frequent variants may have a subtle effect, which we really don't know what that is at the moment. So that's pretty much what I wanted to tell you. I would just come up with summary points about the variants. So we tested about 40 missense variants. 21 had protein structure and everything, and we are fairly confident that now we can assess these variants with this very high accuracy, well, in view of this knowledge and assessment across hundreds of different variants in the group of girls as well as in the population reference databases. The limitations are perhaps the frequent variants, but maybe that's not a limitation, that's a statement of fact. And I believe that this is hugely important in what we're going to do with these variants in the future in terms of treatment management, even for membership in some of these associations, that people can be relatively certain that that's the driver or one of the drivers of the clinical presentations, because a lot of things will unwind from it. Outcome of the study, as I showed it, was that really early onset of the seizures generally predicted poor outcome. I mean, we knew this, but there was no hard evidence for it. So now we have it, at least from that cohort of 271 cases, some of which are related, but they are independent cases. We know, as a statement of fact, this is complex clinical entity, and when I showed you that one single recurrent variant causing pretty much everything from nothing to very severe presentation, I hope that convinced you that that's something, again, we need to keep in mind when we're trying to intervene, because we don't know or predict which way we're going. And I think the PCDH19 is shaping up as the most prevalent genetic seizure disorder in girls. Boys are perhaps under ascertained, because I keep hearing more and more about boys are coming with, especially the somatic variants. We can achieve the variance assessment, and I believe that's going to be important for what is going to be happening next based on all the studies, lovely studies, which we heard today. So I'm only left to say thank you to all of you and the people from my laboratory, especially Yuyan and Christy, who are bolded. Paul and Dan were really long-term collaborators with us, and I haven't showed any really data from the work we do together, but we do work together and actively, but in slightly separate areas of focus. Ingrid Scheffer and Lynette are pediatric neurologists who are greatly contributing. To all of you, and I don't want to dwell on it, you know that you are the drivers, you are the motivator, and you are ultimately the ones we are accountable, and we would like to please you as much as possible with good outcomes. We recently moved, and Paul and I, so our year was slightly interrupted, we moved into new medical school, which is about 6 months procedure to move everything. Physically, it was 2 weeks. In silico and everything, all the approvals, that took about 6 months, but we are in a very nice building, which is on the left. And this is Paul's building, which is almost bigger than this one, but it looks small. And as you can see, we have even koalas supporting the good cause in Australia. Pink noses as of last. Thank you very much for your attention and support.
Questions for Jozef? Very beautiful talk. I want to ask you if in your cohort of patients, the earlier age at epilepsy onset predict also seizure severity and seizure persistence in this patient. Thank you.
I wouldn't want to really comment on it in great detail, because that's a part of the survey and we would like to get additional data. Overall, yes, but if you ask me what type of seizures and what type of clusters, I probably won't be able to give you a very good answer I would be happy with. So it predicts the seizures continue in many of these cases. So it's not only the outcome in terms of the learning and memory problems and regression in that, but of course the seizure activity often persists. So yes, overall yes, but specifically we haven't managed to really pull it apart.
I have a question actually. So now with this new knowledge of low-level mosaicism, I guess would be the best, and then non-penetrance, how as a clinician do you suggest we approach families? Right now it's pretty cookbook, right? Maybe we can have some more genetic counselors to diagnose a child, we check, see either the mom or dad. If it's de novo, we generally stop there. Gets into some of the ethical considerations of testing asymptomatic siblings, either male or female? Do you have any comments on that?
It's very important, Joe, as you know, PCDH19 is not kind of unique in this space of neurodevelopmental disorders. There are many where we have a risk of penetrance is like 8 or 10%, which is even more complicated. Here, you would say that it's highly penetrant. It's because it's 80%, but it's lower than we anticipated. So at least I guess the families generally understand. We as practitioners freak out that we can't really give the 100% correct answer. But I think many families do understand it, that there is a high risk or low risk, if you can stratify it only by that, and that can't be really predicted. But by saying that, we really work hard on biomarker discovery. And maybe we have some clues, but they are probably too preliminary, where at least based on some of the skin cells what we have from penetrant and non-penetrant females is exactly the same variant, we do see differences in certain biomarkers. So that's puzzling me why that would be, but it looks like clearly that maybe there is a way how in the not too distant future we'll be able to predict if a female with a certain variant will be presenting or will be severe or not necessarily severe. So I'm hoping. So this is perhaps a step in between, given the knowledge in the field, but I'm hoping that there will be a better tools to predict the outcome. Because with that comes, of course, intervention.
I guess a follow-up question to that. I mean, a lot of, as a clinician, the way epilepsy syndromes are defined is first phenotypically, and then we look at that group of children and we find the gene. So perhaps, could there actually be a much broader phenotype in patients who have lower levels of mosaicism? Maybe they actually don't have a treatment-resistant epilepsy. Is that possible?
There's multiple answers to that question, yes. Again, we study actively variants which are in the cytoplasmic region, which have not been associated with typical Gecz clustering epilepsy as we know it. And they were found, for example, in males. So these are males with quite a severe neurodevelopmental outcome who had a novel or de novo variation in C-terminal part of protocoherence. Now, because the numbers are like, we have 3 or 4, and we know these variants are functional, given what we know for the time being, it's really, we struggle to say, well, this is another because these males don't reproduce. They are so severely affected. So they can't have girls to experimentally tell us that whether these girls of these boys will be affected even more severely or now not going to be affected. But I have the feeling that these are just different outcomes of loss of or altered function of PCDH19. So yes, there will be other phenotype associated with PCDH19, I believe, and that's simply because we reduce or remove those biases. I mean, biases sounds badly in your expertise because you have to deal in the clinic with many different possibilities. And of course, you remember only so many. And if the diagnosis unwinds from the clinical presentation, of course, there will be a bias, which one you test for genome, which one not, which is affordable, which family can pay for it, where the service can pay for it. But I would say in the future where, everybody is going to be sequenced, or like in some countries in Europe, now they have whole exome, whole genome sequencing on their Medicare benefit, and we're discussing it actively in Australia. It's not the case, but there are proposals for these technologies to be under just a tick box with some gatekeeping, so the money is used responsibly. I'm sure there will be cases which will pop up with slightly different presentation with PCDH19 variation. I'm 100% sure because the makeup of this disorder is that this is broader. So it's hugely important really to get to the bottom of it.
Just a quick question. How do you tell a boy's parents that he has female-limited epilepsy or epilepsy in girls, clustering epilepsy in girls? Are we going to get away from that?
That was a lot of the debate a few years ago. So
I don't know, Joe. You're probably the better one to answer that. I try really not to mingle at it, but the way how, again, it's a term in transition. We just need to accept that the chain is the norm in this field, And I've been in epilepsy for the last probably 25 years since we discovered the first epilepsy gene as such. At that time, there were not that many different epilepsies as you have at these days. So that's the role of the committee and of the fields to really reappraise and change the terminology. So I think for the time being, the girls clustering epilepsy was chosen because it was a bookmark. It was a typical something which distinguished from everything else and made the services aware of this type of presentation. So it started to ring the bell. Of course, then the boys are coming up, so it's not appropriate, fully appropriate. And I guess sooner or later it will change. But I guess as we might reply, maybe you have a better one, Joe.
No, I agree. I think it was a bookmark, and I think the resistance and why there's still heterogeneity in how the terms are being used today is because we want to be sensitive to the males, but we really want there to be greater awareness, right? So as our young epilepsy clinicians are seeing a young girl with clusters, there's the bell, right? And there's not delays in diagnosis, because I think one other interesting thing could be is the age and severity, and maybe we're seeing this in Dravet syndrome. Like, what if we are able to make the diagnosis earlier and treat their clusters more aggressively? Could that, in fact, affect the outcome? We don't know, right? So, Lacey, did you have?
But yeah, as far as comments on the name or other comments asked for, sorry, I zoned out at the
end. Great talk, Joseph. So what was interesting with your cohort study was that the possibility that missense change might be tracking more towards a less severe form of intellectual disability. So I wondered whether you have sort of stratified your cohort into missense versus, knockout, definite knockout patients, maybe the frame shifters, and to see if that correlation tracks further in that sort of broader context. Because we always think about it as being a loss of function mutation. We lump them all in the same basket, but that's in reality probably not correct.
Is asking, it is the case for many other genes and disorders where really it's not just the gene which matters, but it does matter what sort of DNA variant is involved. Here, at least for the time being, we were considering all these variants to be kind of on equal footing, so they contributed kind of in the big picture the same way, destroyed whatever PCDH19 meant to be doing. Here is the first hint, and may not be that simple, but I would say in the scale of things, it's going to be a minor concern overall than as we know it for other disorders, where really it can go from severe to very mild, or really one set of features versus other set of features, or avoiding this or doing that. Here it's going to be minor, but it might be an interesting tweak in terms of interpreting, for example, like the frequent variation, where we see more frequent variants and how they can be contributing to a more subtle phenotype. So that's why we kind of trying to look into it. But I don't think it's of major relevance in its scale comparatively within the PCDH19 missense, nonsense, and these variants. That's at least the statement based on the facts.
Loudly.
With the same variant. No. No, because it's the same variant, so you can't. We don't know how you would mark it as a maternal and affected child, but that's where really this disorder, it's not just a disorder of the variant, it's a disorder of the development. It's a multi-dimensional disorder, and for that we need to have multi-dimensional tools to really understand it and interpret it. That's where I look at it. And that's why we really put a lot of effort into these markers. But clearly the markers for the time being may be working on skin cells, but we can't get skin biopsy for everyone. I mean, I donated mine, and it's not the most pleasant of procedures. It's not really that bad, but that's not something we can request. So we can test the variants, but I think now, based on the tools, we will be able to predict them. So you will not need, we will be submitting this work very soon. I have it in an almost final version. So people will be able to use them. And these tools are not universally good. For different genes, different tools work as it happens, as it is. So you won't need to do a lot of functional study unless you will come across the variant, which is probably in 1 in 50,000 people. You find it in 5 cases in reference databases where you don't know the phenotype, and then you will start thinking, well, then you go, of course, to a step further, and you need to consider different explanations. But I'm hopeful that for the time being, it's going to be simplified by simply clicking it and within 5 minutes having an answer with high probability that this is deleterious, this is not.